Hello everyone. In this presentation, we're still talking about the different types of leukocytes, but now we're going to be focused on the agranulocytes, so the ones that don't present granules. We have already talked about the granulocytes. Now let's focus on the agranulocytes. Therefore, the circulating blood contains two types of agranular leukocytes. These are the monocytes and the lymphocytes. They differ structurally and functionally, and just by looking at these two images, there is going to be a clear difference between the granulocytes that we have covered already and these cells. And this clear difference is easy to see, right? These cells, they show no granules. Their cytoplasm and even their nuclei look clear of granules. First, let's cover the monocytes, which are the largest white blood cells. They are actually 2 to 3 times the diameter of the red blood cells, and we can actually observe this on this image where the pink circles, they will represent the red blood cells. And notice how the monocytes are huge compared to the red blood cells. These cells will account for 2 to 8% of the white blood cell population. Therefore, they're not a very big population. They are normally almost spherical, meaning in a circle. And when flattened in a blood smear, they do appear even larger because they sort of spread out. They are relatively easy to identify by their large size and also the shape of their nucleus. If you notice, these cells, they do have this large oval or even a kidney bean shaped nucleus. Monocytes, they circulate for just a few days before entering the peripheral tissue. And outside the bloodstream, the monocytes are actually going to be called free macrophages, which we can see here in image of a macrophage. And the reason why they're called free macrophages is to be able to distinguish them from the immobile fixed macrophages that are found in many connective tissues. These free macrophages, like the name says, they're going to be highly mobile, and they're also going to be phagocytic cells. And they usually arrive at the site of injury right after the first neutrophil arrives, so they're pretty quick at it. And while they are exerting their function as a phagocyte, the free and fixed macrophages, they will release chemicals that will attract and stimulate other monocytes and other phagocytic cells. In fact, active macrophages, they also secrete substances that will attract fibroblasts into the region. And the fibroblasts, they will begin to produce this dense network of collagen fibers around this site. And this will form a scar tissue that may eventually sort of block off or isolate the injured area. Monocytes are one component of the monocyte macrophage system that includes related cell types such as fixed macrophages, which we talked already about. In this group, we also have specialized cells such as microglia of the central nervous system, the Langerhans cells in the skin, and also phagocytic cells in the liver that are known as Kupffer cells. And then we have also macrophages in the spleen and in the lymph nodes. Therefore, it's a big group of cells with similar functions. Next, we have the lymphocytes, which a typical lymphocyte, they have very little cytoplasm, as we can see over here from this image, with just a thin halo around a relatively large, round, purple staining nucleus. In fact, lymph, the word lymph means clear water, and this is exactly what's represented here on this image. Lymphocytes, they're usually slightly larger than a red blood cell, and they will account for about 20 to 30% of the white blood cell population. So it's a big chunk of the white blood cell population. But remember that neutrophils are the ones that have the greatest population within the white blood cells. Lymphocytes, they're going to be responsible for specific immunity, which is the ability of the body to mount a counterattack against invading pathogens or even foreign proteins on an individual basis. So lymphocytes will respond to such threats in three ways. The first way is where one group of lymphocytes, which are called the T cells, they will enter the peripheral tissue and actually attack foreign cells directly. Another group of lymphocytes are the B cells. The B cells, they actually differentiate into plasmocytes or plasma cells, and they are the ones that will secrete antibodies that attack foreign cells or proteins in distant portions of the body. Now, T cells and B cells, they're so similar that they cannot be distinguished with a light microscope. And the last type of cell are the NK cells. They're called natural killer cells. They are the largest agranular lymphocyte, and they are going to be responsible for the immune surveillance and the destruction of abnormal tissue cells. So they do live up to their name because they go in there and they destroy everything that's not normal to the tissue. And this cell is one of the cells that are important in preventing cancer. So if you have a cell that's mutated and it looks a little bit weird, then the natural killer cells are going to go there and they're going to destroy that cell, preventing the spread and the differentiation and division of this cancer cell. Leukocytes in general, they have a very short lifespan, typically only a few days, and in instances of injury or invasion of an area by a foreign organism, the leukocyte, they will be able to migrate across the endothelial lining of a capillary by squeezing themselves between the adjacent endothelial cells. This process is known as diapedesis. The bloodstream will actually provide this rapid transportation for the white blood cells to these injured sites, where they are going to be attracted to the chemical signs of inflammation or infection in the adjacent interstitial fluid. And this attraction to specific chemical stimuli is called chemotaxis, which draws these leukocytes to invading pathogens, damaged tissues, and white blood cells that already are in the damaged tissue area. You can think of it as a taxi cab filled with chemicals arriving at the injured site. Let's analyze how all these processes work by reviewing the image. Leukocytes present in the blood, they will receive these chemical cues or tips, and they will respond to them by traveling through the blood vessels all the way to the injured site. And once they arrive, they will squeeze their way out of the blood vessels between the endothelial cells in this process that is known as diapedesis or immigration. And once they exit the blood vessel, they will move through the connective tissue of the dermis towards the site of the wound. Some leukocytes, such as eosinophil and neutrophils, they're going to be characterized by these granular leukocytes. They will release their chemicals from their granules that will destroy the pathogen. They're also capable of phagocytosis, so they will clean it all up. And the monocyte, which is an agranular leukocyte, which will then become and differentiate into a macrophage, will also function to phagocyte the pathogen that's present. So they will clean it all up, and once they're done, the tissue will maybe have a scar because the macrophage, remember, they will attract fibroblasts that will deposit collagen fibers and maybe create a scar in that location to be able to isolate it.